All right, everyone, here we are for our last video to go with meiosis. So this is video number 3, and we're going to talk about meiosis 2. So we've been through meiosis 1. We've gone from having 1 cell to having 2 cells. So now we're entering round 2. So now meiosis 2 is very much more like mitosis than the last round. So this is going to look a lot more familiar, but we don't replicate the DNA. Because remember, what we're going for here is for sexual reproduction. So we're going to be combining DNA with another cell, so half coming from one father, half coming from the mother. So you don't want replication at this point. You want each cell to have only half as much as they need. So in some species, meiosis 2 starts right away from just the division in meiosis 1. But in some species, believe it or not, the nuclear envelope reforms and then breaks down again. Not terribly efficient, but that's how it goes. Okay, so prophase 2. So now we're getting those spindle fibers all over again, and the chromosomes are starting to move, starting to head towards the middle. Notice, okay, so down here where it's talking about N, okay, so we are no longer 2N, we are just at N in terms of DNA. So this is now haploid. So metaphase 2, the chromosomes are meeting in the middle. So there's just one set now. In the last round, we had a chromosome and a chromosome. Now it's just, they're all lining up like soldiers, like in mitosis. Single file. Anaphase 2. So we've been attached to these spindle fibers, and the spindle fibers are made of those microtubules, and the microtubules, they are pulling apart these chromosomes. So the sister chromatids are now separating and moving to opposite poles of the cell. Take a minute and look at the colors going on the sister chromatids and how different they are. So this one's solidly purple. This one's got a couple of bands of orange. This orange one has a band of purple. So this is good. This is showing that continued genetic variation, which is what we really want to see coming out of meiosis, genetic variation. So now those sister chromatids are separated and they're moving to the opposite poles of the cell. So anaphase away. They are moving away from each other. And now we have telophase 2 and cytokinesis 2. So the nuclear envelopes are reforming around those chromosomes, going to pack them into the nucleus where they're protected, and the cells divide. But notice we're going from 2 to 4 cells. So the very end result, you end up with 4 cells, and each of these has half the DNA that they need for a complete organism. You get the rest of the DNA from the other germ cell. And that's it. So please notice the variation in colors and the chromosomes going on here, showing all that genetic variation. Notice that these are now all N. So these are haploid cells, and it's all about genetic variation. We want to end up with different organisms. All right, you know where to find me. Send me an email if you have any questions. I'm happy to talk through any of this with you guys, but I want this out there so you can refer to it, since we can't do this in class like usual. Have a great day, everybody.